Couple times.
There it is. All right, good morning, everyone. My name is Cathy Fromen. I'm an assistant professor at University of Delaware in the chemical engineering department, and I started my lab there in 2017. I am one of the Delaware Embry Pilot awardees this past cycle, so I'm just here to give a little overview about my research group and what we're working on, and then to dive into a little bit about what my Embry Pilot project actually is focused on. So as you can see from this opening slide, my group really is into inhalable drug delivery and thinking about the lung specifically as a target for therapeutic intervention. And you can imagine, I mean, especially given the current circumstances, there's lots of reasons to think about how we treat lung and lung diseases more efficiently. So you're probably all familiar with inhalable medicine, inhalable therapeutics. On the left and right of this screen, you can see meter dose inhalers. These are devices that are pressurized where you depress the inhaler, you get a mist that comes out. So this requires timing from the patient. Really the biggest innovation has been these dry powder inhalers. And so this is shown in the middle, an Advair inhaler, where there's a dry powder capsule inside the inhaler device. And this requires the patient to actually breathe, which disperses the powder and allows that to go into the lungs. Inhalable medicines are really good for treating the lung. You can get much higher concentrations of the therapeutic directly to the organ of interest. So there's a real benefit of being able to treat the lung and lung diseases. So just a little background about what is in those inhalers and how we have to design them. There's really precise specifications for what you're going to inhale based on the physics of the lung and the airflow that goes in it. So a particle that you breathe in has to be of a particular particle diameter in order for it to deposit efficiently in the airspace. So I'll draw your attention to the graph on the right. On the x-axis, we have particle dimensions and diameter ranging from 0 to 15 microns. And on the y-axis is our deposition fraction. So if you put in 100% of those particular particle sizes, what percentage would actually land in the lung based on the different regions. And so here you have in red the mouth and throat, that's your upper airways, was before your airways. The yellow is going to be your actual upper airway space. And then often the target area of interest is going to be the deep regions of your lung, alveolar regions, which are shown in purple. And so really the conventional wisdom, if you want anything to get to the lung, it's between 1 and 10 microns. If you want deeper regions, you need to be in the 1 to 5 micron range. And you'll note there is a little tick up in these like very sub nanometer size aerosols as well. But this is really a pretty narrow size range when we think about engineering particles and how to actually manufacture these things in a very particular size controlled range. So once you inhale these, though, there's a lot more challenges. So not only do you have to get it out of a particular particle dimension, but you have to consider the microenvironment that it's landing into. So here I'm showing a particle and what may happen to it once it reaches these deep regions of the lung. So your upper airways, and as you go lower and lower, there's a mucosal lining that covers all of your airspace. This is actually a dynamic layer that is constantly moving upward out of your lung. So anything that gets deposited is trapped in your mucus, and then that mucus layer is moved upwards. So this is obviously a physical challenge as you think about it's meant to protect you from various infections, but also a challenge as we think of delivering therapeutics. So other challenges are going to be then the cellular microenvironment there. And so your deep lung has a whole host of macrophage cells. So these are essentially garbage collectors. They roam around and pick up particles and foreign objects. And the main job is to sequester them and to then respond to any insult. So I have a video here of macrophages eating foreign particles. These are synthetic particles that we made in the lab, so that's why they have the little donut shape. They're pink when they get inside the cell, and they're blue when they are outside the cell. And you can see these macrophages are roaming around. They're not only picking up particles, but they're eating dead cells. And so they're able to not only sequester these foreign objects, but they're also responsive to their environment. They can regulate the local immune response by putting off cytokines and signaling to other cells based on what they're eating effectively. And so macrophages are essentially garbage collectors. They're there more for protection, but there's other subsets of these innate immune cells, these phagocytic cells, including dendritic cells, that can also sample the microenvironment and respond accordingly by mounting antigen-specific responses and initiating the adaptive immune response. So the lung is a pretty complicated environment, and again, I mentioned these really narrow particle size distributions. But if we look at conventional inhalers, we zoom into what the particles look like and what the powders look like, and essentially they're very heterogeneous. They're made by either a micronizing or spray drying process that yields very non-homogeneous particle sizes, shapes, morphologies, all of these things that our cells are going to be able to sense and respond to that we're not controlling at all from the upstream inhaler design whatsoever. Part of this, if we look, is kind of commercially driven. So if we look at the top list of inhalable therapeutics, you don't have to care so much about the details to know that most of them are for asthma therapy. These are going to be small molecule drugs that are very potent, and they require very low doses to be somewhat effective, and they can afford very low efficiency. So there's all sorts of stats about, anywhere from 10 to 30% of the dose is actually reaching the airspace, depending on how well patients use this. But these are very inexpensive for pharmaceutical companies to make, and yet they're making tens of billions of dollars in inhalable pharmaceutics. So it's very difficult to break into this market because we have this standard of, we make these kind of crappy powders and crappy devices that are very inefficient, but they work well enough and we're making money on it. So there's a hard way to break into the market and think about optimizing. But you can imagine lots of lung-specific diseases that would really benefit from changes to our therapeutic paradigm. Something like tuberculosis that's going to have very specific lung infections, getting high drug concentrations directly there and directly to the site of infection is really important and could really change the way we're treating these diseases. And so I could go through all of these as examples, but there's really pulmonary diseases, and currently, our current crisis, we need better ways to think about treating lung diseases. So if I had to summarize the need, really we need to understand better what happens on the particle lands, what is it interfacing with, how does your therapeutic as a function, how you design your therapeutic actually change the response in the lung, and then how can we better design our therapeutics so that they're giving really coordinated, complicated molecular cues. Maybe these are monoclonal antibodies, maybe these are immune engineering responses to be vaccine oriented or some other way to direct the immune response. And then how do we make these more efficient devices that actually get to where we need them to go to and work the way we need them to. So this is what my lab focuses on. We're a chemical engineering lab, and so that has a lot, we think about aerodynamics, we think about immune engineering, which requires us to know about the mucosal immunology, and then we're able to design new materials at that interface to drive new responses. And so our lab really has kind of two divisions to it. The one that I'll talk about today is how we think about designing our therapeutics, what they have to look like, how is the chemistry oriented at the surface so they can control and direct immune responses in the lung. That has broad implications to lots of different diseases. And the other thing we're focusing on, which I won't talk about today, is that we're designing these large, full-scale, three-dimensional models that look like a human lung. And so we're making these breathe, and we're putting aerosols into them and trying to understand how the inhaler design can be used to change where the drug is going based on how the patient is breathing. And so we've got some interesting things scaling lungs down to pediatrics and various disease states with that. But I won't talk about that today. I'm just going to talk about our work really thinking about the immune environment of the lung and how we design therapeutics for it. So I have 2 great graduate students at the top who are working on this project, and 3 undergrad students who have had their hands in some of the aspects I'll talk about today. I won't spend a lot of time on this, but essentially we have 2 platforms that we're working with where we can design very homogeneous inhalable formulations that are we're able to really control at the molecular level the spatial chemistry of the therapeutic itself. And so we have two carriers. One is a metal organic framework-based nanoparticle where we can really control the exact spacing between molecules. And so we're looking to see how that spacing can be used to direct immune responses. But the one I want to talk about today is our work with polyethylene glycol diacrylate particles. And so it's sort of a mouthful, but the big thing to take away is they're made of PEG. And PEG is considered to be an inert substance, an inert polymer that is used to functionalize all sorts of therapeutics. It's put on to monoclonal antibodies to increase their circulation time. And so this is thought to be relatively inert. Now it's also in your shampoo, for instance. There's growing literature to say that might not necessarily be true, but it's something that's definitely FDA approved at this moment. So these are supposed to be non-stimulatory, inert materials that we can use to then add in different chemistries and see how our cells can respond. But we've noticed something very interesting with these base inert particles on their own that I'll share with you today. So we can take primary macrophages out of the lung or from other sources in a mouse, and we can culture these. And so now primary macrophages are sort of difficult to work with because they're primary cell cultures. Eventually they're going to die if you don't give them some sort of growth factor. And this is true for sure of macrophages. And so we wanted to just check if the same, how these cells are responding to our inert material or inert particles as a baseline. And we found something very interesting. So in the top panel, these are untreated cells, and you can see from day 1 to day 35, our primary macrophages, they're all dead. And in our hands, they die from anywhere from 2 to 3 weeks, depending on a number of factors. But you'll notice in the bottom, day 35, these are our treated particles given with a relatively low dose of these inert, non-stimulatory therapeutic particles that we're seeing a lot of cells that are still alive. If we take this out to 6 months, we actually do still see cells that are maintaining culture, they're still active, and they're still responsive to taking up new material. And so this is really remarkable because this is a single administration of particles, and it has effects on that cell viability that spans over a 6-month lifespan. Now, this was really surprising to us, and we're engineers, and so we're not immunologists, but we're truly trying to figure out what is going on and how we can use this effect to think about how we change therapeutic approaches. So the first thing that we wanted to test, which maybe is obvious to anyone who's worked with the macrophages, are they activated? And so a macrophage is known to take up particles. This is not a surprise, but usually they respond in some sort of activated manner. Typically that involves either becoming a pro-inflammatory or let's say anti-tumor type of macrophage, or they can become an anti-inflammatory activated state or an M2-like or pro-tumor pro-wound healing sort of cell. But regardless, they have to be activated. And I'm just showing examples here, but we don't see any signs of activation with these cells. And that's really probably the most striking aspect of it. Not only are they living forever, or they're living for a much greater amount of time, but they're not activated in the traditional sense as if they had eaten a bacteria or they'd been responsive to a fibrotic environment or cancer environment. What we do find is that they are delaying apoptosis, and so we can do this by a number of different mechanisms. What I'm showing here is just the Nextin-5 staining that looks at phosphatidylserine expression on our cells 1 day after eating the particles. And so our untreated cells are undergoing apoptosis or programmed cell death, and treating them with particles is basically reducing this apoptotic response. And so now we've spent a lot of time trying to figure out what is really going on and how are these changes happening. And we just recently got RNA sequencing data back that shows there's very little changes between our treated and untreated cells. We've identified about 300 genes that are different, but those changes are less than twofold between our treated and untreated cells. So we're starting to pick apart our pathway, but it really starts at the lysosome, and we can identify some upregulated cells in the ERK and AKT pathways that we think are responsible for this enhanced survival, but not going through kind of the normal traditional macrophage activation pathways. So we wanted to think of how we could use this for a benefit. So not only are we trying to figure out what's going on, but how is this important and does this happen in vivo and all these sorts of things. And so this is the focus of my IMBRI pilot project, is thinking about how we can use these long-living macrophage cells as a new cancer therapeutic. And so if we think of kind of the most cutting-edge cancer therapies, we've seen this explosion in CAR T. So these are T cells we take out of a person, we stimulate them, we give them antigen specificity, and we put them back into the body. So macrophages could be something even more effective than our CAR T approach, but that same kind of take them out, put them back in. But macrophages could be great because they're antigen agnostic, and so they wouldn't require you to have, here's a tumor specifically, but they could kind of go into the tumor microenvironment, sense what's wrong, and they're not, they haven't been exposed to the constant tumor suppression that like the in situ macrophages would have. And so really, this has been tried in a number of cases, but they have really poor survival upon reinjection. So that was our first hint, hey, we have this way to keep macrophages alive longer. Could this be a way to increase macrophage survival upon reinjection? And also thinking about keeping them from repolarizing into a pro-tumor type of phenotype. And so this is really the concept that we're pursuing with this project. The idea is that we would take essentially monocytes from the blood, we differentiate them to macrophages, and we polarize them into an M1 or an anti-tumor phenotype. We'd give them particles ex vivo, and then we would reintroduce these now long-surviving macrophages into the body. And the idea is that they would be able to go into the tumor, they'd be in this M1 long-surviving anti-tumor environment that would be able to sense the tumor and work on destroying it. And so that's the concept of the project. We really haven't gotten that far yet. We're trying to establish our lung cancer model, and so this will be in an orthotopic model that we're looking to do the reintroduction, so putting these back into the lung. Yeah, that's really where we're going at. So we're excited to get this started. We certainly are spending a lot of time figuring out what is going on with these macrophages after they have injected particles. And so this is just my group. I'd like to thank all my collaborators who've helped on this, especially my funding, the Delaware IMBRI for being able to pursue this kind of new direction in cancer therapeutics and lung cancer specifically. And with that, I will stop and I'm happy to take any questions.